The most general phases of the cell cycle are interphase and mitosis, where interphase is basically growth of the cell, and mitosis is its division. Let's start with interphase, which itself has three parts, G1, S, and G2. So what does a cell need to do during interphase? One of the main limiting factors of cell division is how fast it can grow, since it can't divide until it gets big enough. And what limits how fast a cell can grow? Mostly the amount of ribosomes it has, since cell mass is mostly dependent on the abundance of proteins, and proteins are made by ribosomes. This growth happens during G1, which is the longest phase of the cell cycle. This is also the only time that cells can exit the cell cycle to enter G0 phase, in which they are not dividing. Some cells in G0 are still capable of dividing when stimulated to do so, and can re-enter the cell cycle in G1, such as fibroblasts, hepatocytes, and lymphocytes like this one here. Cells can also leave the cell cycle during G1 to become terminally differentiated, such as neurons like these, or muscle cells or erythrocytes, which will never divide again. Once cells transition into S phase, they're committed to complete the cycle, so S, G2, and mitosis progress much more rapidly. What's an example of a tissue that has cells that are constantly proliferating? There's a bunch of examples, but one is the skin. And by the way, these cells that are constantly dividing will be hardest hit by anything that interferes with cell division, like chemotherapy. Okay, what happens during S phase? S stands for synthesis, and this is when DNA duplication occurs. Therefore, cells in G2 have twice as much DNA as cells in G1. In both cases, they have 46 chromosomes, which is 23 pairs of homologous chromosomes, or 46 chromosomes total. But in G2 phase, each chromosome is made of two sister chromatids, whereas in G1, each chromosome only has one chromatid. Mitosis is the shortest phase of the cell cycle and includes prophase, metaphase, anaphase, and telophase. This is when the chromosomes condense and separate, and one cell becomes two. After mitosis and cytokinesis, the single parent cell is now two daughter cells. So let's talk about regulation of the cell cycle. What happens if a cell cycle is not regulated properly? Well, this can cause cancer. Cells can become neoplastic and form tumors. Therefore, it's really important that there are checkpoints that control transitions between different phases of the cell cycle, so if something is wrong and the cell should not be dividing, it can put on the brakes. One family of proteins that regulate the cell cycle are the cyclins, and the cyclin-dependent kinases. Cyclin-dependent kinases are constitutive, which means they're always around in all phases of the cell cycle, but sometimes they're active and sometimes they're not. So what activates them? This would be the cyclins, which is why they're called cyclin-dependent kinases. The concentration of cyclins fluctuates over time, depending which phase of the cell cycle the cell is in. There are several different cyclins and they're present at different phases of the cell cycle. The cyclins will activate specific CDKs, and the CDKs will then activate other proteins to do whatever needs to be done during that phase of the cell cycle. For example, in S phase, they'll activate proteins to initiate DNA replication. By the way, as a preview to a later part of biochem, what does the kinase and cyclin-dependent kinase mean? This means that they phosphorylate their targets, and phosphorylation is often used to activate or deactivate proteins. Since the G1 phase can last indefinitely, but once cells transition to S phase, they're committed to divide, the most important transition to regulate is the G1 to S phase transition. This is why tumor suppressor genes, such as retinoblastoma and p53, are both able to prevent this transition. So what's a tumor suppressor gene? It's a gene whose protein product has a role in preventing cell division, and thus plays a major role in suppressing tumors. Okay, so what might happen to a cell that would make it want to halt the cell cycle? One of the most important things is DNA damage. p53 is one of the most notorious genes for detecting and responding to DNA damage, which has earned it the nickname the guardian of the genome. If p53 notices something wrong with the DNA, it'll prevent the cell from dividing, thus preventing tumors from occurring. When DNA damage is detected, p53 will stop the cell cycle and initiate DNA repair pathways to try to fix the damage. If the cell is able to repair the damage, the cell cycle can restart. However, if it can't, p53 will initiate apoptosis, because it's better for a cell to die than to risk developing cancer. Retinoblastoma, or RB, also prevents the transition from G1 to S phase, but works a bit differently. Basically, RB has a few sites that can be phosphorylated, but after mitosis ends, RB loses most of its phosphate groups. In this hypophosphorylated state, it is active and it's suppressing the G1 to S phase transition. Only when it's inactivated by adding more phosphate groups will it allow the cell cycle to continue. And guess what type of protein adds phosphate groups to RB to allow the cell cycle to continue? That's our friend the cyclin-dependent kinase. Tumor suppressor genes like RB and P53 follow the two-hit rule, which means that both copies of the tumor suppressor gene have to be mutated to allow neoplastic transformation. This is because if only one copy is mutated, the other one is sufficient to regulate the cell cycle and prevent tumorigenesis. By the way, P53 is so powerful that more than half of all cancers have loss-of-function mutations in P53. Okay, we're going to switch gears now and talk about the different parts of the cell. We'll start with the rough endoplasmic reticulum, or RER. The rough endoplasmic reticulum is endoplasmic reticulum that's covered with ribosomes. This gives it a rough appearance under electron microscopy. Here's a scanning electron micrograph showing some rough ER, and I want to point out that it's generally found near the nucleus, which is this round object here. But it's hard to appreciate the detail from this, so here's an artist's rendering of some more rough ER, and you can see that the smooth ER has no ribosomes, and the rough ER is covered in it. So why would you want ribosomes to be in this membranous structure instead of floating around the cytoplasm? Because the rough ER is where secretory proteins, which are destined to be exported from the cell, and proteins that will end up embedded in the membrane, are synthesized, and it's more efficient to make them all in an organized location instead of trying to round them up later, after they've been in the cytoplasm. The rough ER is also the only place where certain post-translational modifications are added. Can you think of any examples? The main one is N-linked glycosylation, in which a group of sugars, which is called an oligosaccharide, is added to the nitrogen atom of an asparagine amino acid on the protein. So here's our asparagine, and here's the amine and carboxyl groups that all amino acids have. And here's the part that makes asparagine unique, which includes the nitrogen that gets sugars added to it. Why is it called N-linked glycosylation? Because we're adding the sugars to a nitrogen. But you can also use this to help you remember that you're attaching it to asparagine, since this amino acid is represented by the letter N. Okay, so what types of cells do you think might have lots of rough ER? This would be any cell that secretes lots of proteins, so some examples are mucus-producing goblet cells and antibody-secreting plasma cells, like the ones you see here, with their eccentric nuclei to make room for rough ER. And don't forget about neurons. We don't often think about neurons as secretory cells, at least I don't, but in fact they make tons of vesicles filled with peptide neurotransmitters, which they're secreting all the time to send a signal to the next neuron. And by the way, in a neuron, the rough ER is called a Nissl body. Okay, what about the smooth endoplasmic reticulum? How is this different from the rough ER? Well, the smooth ER does not have ribosomes attached to it and has no role in protein synthesis, but it has two main functions, which are steroid synthesis and detoxification of drugs and poisons. What cells produce lots of steroids and therefore have lots of smooth ER? This could be hepatocytes, gonads, and the adrenal cortex. And what about detoxification of poisons? That would also be hepatocytes. There's one more thing you should know about the smooth ER. This is where the protein glucose 6-phosphatase is located. What does glucose 6-phosphatase do? It catalyzes the last step in gluconeogenesis by removing the phosphate from glucose 6-phosphate, which allows it to be exported from the cell. Do you remember the name of the disease that's caused by glucose 6-phosphatase deficiency? This is von Gierke's disease. Alright, time for our first flash quiz of the cellular section. The tumor suppressor genes P53 and RB prevent the transition from the blank phase of the cell cycle to the blank phase. So these guys block the G1 to S phase transition, because remember, once you get past that step, the cell is pretty much committed to go through the rest of the cell cycle. Now let's go over cell trafficking, or how proteins are sent to one place or another in the cell. The main character here is the Golgi apparatus, which is kind of like the specialized endoplasmic reticulum, which has some modifications for packaging molecules before they're exported to their final destination. This is a scanning electron micrograph of a Golgi apparatus. Honestly, you'll probably never need to be able to recognize this on your exam, but just in case, you can see here that the Golgi is pretty easy to recognize. It basically looks like a cross-section through a stack of pancakes, and sometimes you can see these vesicles coming off of it. As I mentioned earlier, N-linked glycosylation occurs in the rough ER. However, these N-linked oligosaccharides can be further modified in the Golgi. Also, O-linked glycosylation occurs exclusively in the Golgi, and only occurs on serine and threonine residues. So what's O-linked glycosylation? It's pretty similar to N-linked, except now we're adding the oligosaccharides to an oxygen atom, which is on a serine or a threonine amino acid, which is why it's called O-linked. So like before, these are the amine and carboxyl groups that all amino acids have, and here are the oxygen atoms that the O-link glycosylation will add sugars to. So like I said before, the Golgi is mostly known as the distribution center of the cell. Now how does it know what needs to be sent where? Well, specific modifications on proteins can target them to different organelles, and the most important example of this for your exam is that adding mannose-6-phosphate to a protein targets it to the lysosome. So here's mannose-6-phosphate, which is just a mannose sugar with a phosphate group on its 1, 2, 3, 4, 5, 6th carbon. Now if someone is unable to add mannose-6-phosphate to a protein, where are those proteins going to end up instead of the lysosome? Well, they'll be secreted outside the cell, and this results in a disease called eye cell disease. How can you diagnose this? Well, if you see high levels of lysosomal enzymes in the plasma, that's a big clue. Patients with eye cell disease will often have coarse facial features, clouded corneas, and restricted joint movement, and this is usually fatal in childhood. This might show up on a test as a really sick baby with lab results showing lysosomal enzymes in the baby's blood, and they ask you which of the following mechanisms is responsible for the disease. And of course you'll pick the one about not being able to add a sugar to a protein. Now not only can the Golgi add sugars to proteins, but it's also important for assembling proteoglycans, which are proteins that have lots of polysaccharide chains attached to them. These are often secreted into the extracellular matrix and used as a cushioning filler substance. So where do you think you might find it? Well, there's lots of proteoglycans in cartilage and in joints, and one of the main ones is chondroitin sulfate, which as you can tell from the chondro in the name, like chondrocyte, is an important part of cartilage. And people even take this as a dietary supplement for osteoarthritis, although I think the jury is still out on whether or not it actually helps. Lastly, the Golgi is where many post-translational modifications are added to both proteins and proteoglycans, such as sulfation of tyrosines on some proteins. So now let's talk a bit more about vesicular transport to and from the Golgi. The Golgi is found between the rough ER and the cell membrane. And the cis Golgi is what we call the side close to the rough ER, and the trans Golgi is what we call the side close to the plasma membrane. There are three main proteins that organize traffic to and from the Golgi, as well as between the different layers. COP1, which stands for coat protein 1, is involved in retrograde transport from the Golgi back towards the ER. It's also used to bring things from the trans Golgi back towards the cis Golgi. So basically everything in this downward direction. Now COP2, or coat protein 2, does just the opposite. This causes anterograde transport from the rough ER towards the Golgi, although this one does not have a role in transport from one part of the Golgi to the other, like COP1 did. Now you might have your own way of remembering these, but I like to think of it as 1 being symbolic of the beginning, like the starting point, so COP1 goes towards the beginning, which in this case is the rough ER, and COP2 goes the other way. Lastly, clathrin is important for receptor-mediated endocytosis, and is also used to send vesicles to lysosomes. When something is endocytosed from the extracellular environment, it's first stored and handled in a temporary organelle called an endosome. Depending on what the endosome contains, it can be sent to a lysosome for degradation, or to the Golgi for more processing, or even back to the membrane for excretion. Peroxisomes are the organelles where catabolism of very long-chain fatty acids, like this one, begins through the process of beta oxidation. This converts them into medium-chain fatty acids by removing pairs of carbons as acetyl coenzyme A. And the medium-chain fatty acids are then transported into mitochondria and completely degraded, which will be discussed more later in the chapter. So if a patient has dysfunctional peroxisomes, what happens? The main problem is that long-chain fatty acids accumulate, both in tissues and in blood, and can cause neurologic defects. This is called Zellweger syndrome, and it's autosomal recessive. These patients will have defects in neuronal migration and hypomyelination by the time they're born, and after birth can lose hearing and vision. So how would you recognize this in a newborn? Hypotonia, seizures, and an inability to eat would be the first clues that something's wrong, and hopefully after further testing you'd see that very long-chain fatty acids accumulating. The proteasome is a large complex of proteins whose function is to degrade other proteins into small fragments about 8 amino acids long. This is a very important function, since misfolded or damaged proteins can cause disease, such as the beta-amyloid proteins in Alzheimer's disease. Proteasomes look kind of like a barrel and have a regulatory component shown in red and a catalytic part shown in blue. Since you don't want these things going around degrading all your proteins, cells have a system that allows the proteasome to be selective. So here's a protein that in some cases we might not want to have around, so we want to degrade it. This is the protein huntingtin, which aggregates and causes Huntington's disease. Proteins that need to be degraded are recognized and then modified by enzymes called ubiquitin ligases, which covalently attach a small protein called ubiquitin to the target protein. Ubiquitin binds to the proteasome's cap, which causes the barrel to open and allow the protein inside, where it is degraded. For our next flash quiz, can you tell me where a protein is going to end up if mannose-6-phosphate is attached to it in the Golgi? That would be in the lysosome, and remember, if this mechanism is defective, you can have eye cell disease.